Okay, so let me just share the slide. So we're continuing at where we stopped on lecture 5. That's our final lecture under my own section. So we looked at fermentation. We started with this fermentation in the last class, and we saw the different types of fermentation processes, the microbial and the enzymatic. And so we also looked at economic considerations of microbial fermentation. We looked at that, and then we were at the conditions or the factors that affect a fermentation yield. Looked at the conditions, the factors that affect fermentation yield. That was the last topic we're looking at. And under that, we looked at the inoculum, the amount of inoculum. We looked at the initial level of contamination. We looked at the pH level. Those are the factors that affect fermentation yield. And we were looking at temperature when our time was up. So temperature is another factor that affects the activity of microorganisms. So to get them to work at the optimal activity, we have to give them the best temperature. I think I have to also still be admitting people, so that will be distracting me again. Okay, so we have to give the best temperature to the microorganisms for their optimal activities. So for microorganisms that grow naturally in soil, water, and air, those ones work best at the temperature between 25 to 30 degrees centigrade. And those that are isolated from animals, they grow best at 37 degrees centigrade. Microorganisms that have their growth favored by low temperatures are referred to as mesophilic. And those that thrive best at higher temperatures are known as thermophilic. So it's very important to study the temperature tolerance of the microorganism of your concern or your interest, so that you can put them at that best temperature for them to work well. Okay. So during fermentation process, heat is produced. So it's important to control the temperature so that you maintain the exact temperature that you know that is going to be suitable for the growth of the microorganism of your concern. So another factor is product concentration. The product concentration may increase to the level that, sorry, I've skipped. It may increase to the level that inhibits the activity of microorganisms. That is, the microorganisms are useful for us in this case. So product concentration might increase to the point that it can inhibit the microorganism activities. So that's another factor. So when the activity is inhibited, it means it's no longer yielding. So another factor is initial concentration of the raw material. Raw material is what we call substrate. Example is sugar. It may be too high and then adversely affecting the microorganisms. Just give me a little time. Let me admit more people. Okay. So another factor is oxygen. Some microorganisms are aerobic and some are anaerobic. So those that are aerobic thrive in the presence of oxygen are fungi, algae, and some few bacteria, while some few bacteria are anaerobic. Some bacteria are anaerobic, some are aerobic. So if you have low oxygen for aerobic microorganisms, that will lead to poisoning because they don't have enough. And if you have too much oxygen, it will help the microbes to excessively grow at the expense of the product. Instead of producing the product you're looking for, they will be growing. So that's another adverse effect. So it shouldn't be too low, it shouldn't be too high. Then another factor that affects fermentation yield is level of nutrients in the substrates. May be sufficient, may not be. So these nutrients include starch or sugar. The cell of microbes, they contain 50% carbon. And so the carbon source for the growth is important. So we have sugar, that's starch, we have sugar, we have carbon dioxide, all such as are required. And the cell mass also contains some nutrients like nitrogen. So the source of nitrogen for this is ammonia or any ammonium salt, for instance, diammonium phosphate. Also, amino acids and some vitamins are also important. We also need to include some minerals like phosphorus, potassium, and so on. So one key thing that is important is that you have to keep the environment, you have to keep fermentation process very, very sterile. Maintain sterile condition while carrying out the fermentation process. You might be wondering, why do we have to keep it sterile when it is microorganisms we're looking for to use in this? It's not all the microorganisms. You have all microorganisms, they will all eat up your substrates, your raw material, and they're producing different things. It's not what you're looking for. So we're selective of which microorganisms that we are using. And so the different microorganisms, as you have seen, require different conditions for them to thrive well. And so that is why it's very important to have sterile condition while carrying out fermentation process to avoid low yield and to avoid loss of culture. The culture is where the microbes are, microbes culture. So to avoid the loss of the microbes culture and to avoid low yield, you have to carry out the sterilization. Sterilization can be carried out either in batch or continuous mode. And you have to sterilize the air, carry out the sterilization 2 types. Sterilizing the air, because we're going to use some air, and also sterilizing the medium, the microbes medium. So in this case, we're looking at how we sterilize the medium or the media case of many of them. So to sterilize the media, these are the steps that you must follow. You purge steam into the medium until the desired temperature is reached. Of course, you will study your microbe very well and know this is the temperature it will thrive. And then other ones might not thrive there. So you purge steam into the medium until you reach that desired temperature. Also, you can use electric elements to heat the medium. You can also use jacketed vessel to pass steam through the jacketed vessel. That is the vessel where the fermentation is taking place is covered with a jacket. So it has another level of metal covering it. So you pass the steam through the jacket to raise the temperature during sterilization. Of course, you know that during the fermentation, temperature rises. So it's important also to carry out some cooling just to bring the temperature to the optimum temperature for the microorganism of interest. Okay. And you also have to sterilize air. I've told you, you sterilize the medium and you sterilize the air. So there are very many airborne microorganisms all over the place. So, and some of these absorb in airborne dust, and then they can easily move in along with the air that you're using for the fermentation. So in this case, you have to use some filters. You filter the air particle before you introduce the air into the fermentation process. Okay. And then in the lab, if you're carrying out this process in the lab, you can use cotton plugs to block where the air is coming in so that air that you are putting in will be filtered. If you're carrying out just a pilot plant or a small scale fermentation process, you can use small fibrous filters to block so that the air that you're introducing in will be filtered to not carry all the microbes inside. And then you not carry any microbe inside, because the only microorganism that is supposed to be there is the one that you purposely put there by yourself. So any other one coming, you block from it coming in. And then also, if it is a large industrial fermentation process, in the large fermenters, heat may be used to sterilize the air. Okay, so air compressor can be used to raise the temperature to the level which will kill the microbes that are not what you want to kill. Any microbe, and then you introduce sterile air into your fermentation vessel. So the steps we have to take in fermentation process, you have to, first of all, you preserve your culture. The culture is the microorganism that you culture, that you grow. So you have to preserve it. So a preserved high-yielding microorganism strain is a prerequisite of any good fermentation process. So this preserved culture of your microorganism of interest, what you're using for the fermentation, the culture that is preserved, you will just pick a little to put into the fermentation vessel. That is the inoculum. That is the initial cells that you put. It's a little that you use. And then from there, you will scale it up to whatever size that you need. And those microbes, the preserved microbes, the preserved stock is always in the form of an inert spore. Inert spore is not active, it's just inert. So the next one is you scale up. So scale up is an important part. That is scaling up the amount of the microbes. You scale it up in your fermentation vessel. So for some fermentations, it's essential to amplify the starting inoculum. You can see in the number 1 here, say just a little of it is what you use to initiate the process. Then after that, you amplify it, increase in volume enough for the material for it to be able to carry out the fermentation you want. So, and in bacteria and yeast systems, where vigorous growth is anticipated, the scaling up you will do is you scale up to an initial to final inoculum ratio. The initial one is the small one, the little one you added. You can scale it up to final inoculum. So you scale up from 1 to 20 ratio. That is, 1 is initial, 20 is final. You scale 1 to 20 ratio to 1 to 100 ratio. This is what is the common scaling up that is carried out. So initial 1 is to final 20. You scale that up to 1 to 100. So, and in this case, what is used to generate and produce the microbes that we use is called seed tanks. The seed tanks are designed to provide an ideal environment for growing microorganisms. In the seed tanks, you fill the seed tanks with the things that a particular microorganism will need to survive and thrive, including warm water. You add carbohydrate, which is a food like lactose or glucose, sugar. Then you add necessary carbon source like acetic acid, alcohol, hydrocarbons, and nitrogen source like ammonium salt. You add all those in the seed tank. Give it the best condition for it to grow. So vitamins, amino acids, and minor nutrients also, it can be added to the seed tank. And then the seed tank is equipped with mixers where you have to mix the medium very well. And then the air that you're passing into the medium needs to be delivered sterilized. Filtered air, like I mentioned to you before, that you have to sterilize the air that is going to be used. 2 sterilizations, you sterilize the medium, sterilize the air. So the air you're putting in should be sterile. So after about 20 to 28 hours, then the material in the seed tank is transferred to the primary fermentation tank. So you produce the inoculum, everything in the seed tank, you do everything and add all the necessary conditions and keep it between 24 to 28 hours. Then it is ready. So you now carry it and introduce the amount you're introducing into your fermentation tank. The fermentation tank is essentially a larger version of your seed tank. Everything you have in your seed tank is what you have in your fermentation tank, but in larger quantity. So here, the microbes are allowed to grow and multiply. During this time that they're growing and multiplying, with everything you put there, the nutrients, the substrate, everything, they will start excreting the quantity of the products that you desire. As they multiply, in that fermentation tank, they'll start excreting those products that you desire. So then the tanks are cooled, the fermentation tanks are cooled to maintain the right temperature. So, and it's also, apart from the cooling to maintain the right temperature, it's constantly agitated, constantly stirred. And then the continuous air stream of sterilized air is pumped in continuously, pump in sterilized air. And then also anti-foaming agent is periodically added so that, to inhibit foaming. Acids, you can also use acids or base to help to control the pH so that it will be at the particular pH that your microorganism will grow optimally. And then you make sure that aseptic operation is carried out. So for this to happen, that is the operation which is free of any disease-causing microorganism. So this is what should be carried out at prolonged time period. So in this case, to achieve this, you have to make sure that you expose the pipeline that is transporting the sterile air, that is transporting the seed, that is transporting other materials, that is transporting the seeds, the sterile air, and other materials that are required for the fermentation into the fermentation tank. You make sure that those pipelines are sterilized. They are exposed to steam, usually at 120 degrees centigrade for 20 to 30 minutes. So this is the schematic diagram for what I'm just explaining to you. This is how it looks like. When I give you the notes, you can go and look at it, but I've already just explained everything about this. This is your fermentation tank. Okay, now after the fermentation, the products, everything with the microbes and the cells, everything is modeled up inside the tank. So it's then important for you to separate your product and purify your product. So recovery and purification of fermentation products, it can even be much more difficult and more expensive than the fermentation step itself. So certain products are contained in the cells that excrete the product. Sometimes the products are even inside within the cell. It's already produced, but it's within the microbe cells. And so they're not released, or sometimes they just partially released into that mixture that we have. So it's in case you have to break the cells to release the product, it may be possible or necessary to wash the cells to remove impurities first before breaking the cells to get your product out. Some cells have high resistance to shear, to tear them, they resist. So in this case, they require specialized equipment to rupture them. So we have hydrodesulfurized products, blended or used as catalytic reforming feedstocks. That is from the fermentation process. So sometimes you might need ultrasonic vibrations to be able to, and grinding mills to rupture the cells of your microorganisms. You also might need high pressure to rupture these cells. Centrifuge, rotary drum filtering are commonly used in the separation of the cells from the broth. The broth is a mixture of everything, the microbes, the products, everything together in the cells. So you can use centrifuge, you can use rotary drum filtering. These are the commonly used methods to separate the cells from the broth. So products in the broth may be recovered by a variety of unit operations. The products from that mixture can be recovered by different methods. One can be distillation or extraction with salts and solvents, membrane ultrafiltration, precipitation, and also chromatography. So that is it with microbial fermentation. Now we'll just look briefly at the enzymatic fermentation using enzymes. Enzymes are proteins. They occur naturally in microorganisms. They occur naturally in animal organs, and they occur in vegetable extracts. They are biocatalysts. They bring about specific biochemical reactions without their structures or their quantity being changed. Their activity depends on certain factors, depend on temperature, pH, storage time, and storage conditions. For enzymes are very specific to the particular reactions that they catalyze. And so they are specific and they will just produce a particular product, just one product from the selected enzyme that is used. And so because of this, the purification of the product in this case is less expensive, unlike that of microbial fermentation, because microbes can produce several products, everything mixed in, and you have to do a thorough separation. So even though enzymatic fermentation it's easy to separate the product, enzymes themselves are more expensive than microorganisms. So either way, you will still make expenses. So either you're using microbes or you're using enzymes. So now let's look at manufacture of fermentation alcohol. Manufacture of alcohol, ethanol, by fermentation. So biomass materials that are used in this case are those that contain sugar, starch, and cellulose. The sugar-containing raw materials include sugarcane, sweet sorghum, sweet potatoes, sugar beets, and molasses. We also have cassava as a very viable starch-containing material, which is tapioca. We have maize and potatoes. So these materials contain disaccharides, which are easily hydrolyzed by water to reducing sugar like glucose and fructose, like you see in this equation below. So from sucrose, hydrolyze to glucose and fructose. So before you carry out the fermentation, the starch is hydrolyzed to polysaccharides and then to saccharified and then saccharified to glucose. The cellulose contains complex carbohydrates, and so they require heat and enzymatic treatment to extract the fermentable polysaccharides. The cellulosic raw materials include straw and bagasse. Microorganisms mostly employed in alcoholic fermentation are yeasts. So glucose is fermented into alcohol according to this equation you see here, using yeast. It's fermented to alcohol, carbon dioxide. So there are several viable yeast strains that can be used, but the most commonly used strain of yeasts that is used for alcohol or ethanol fermentation is Saccharomyces cerevisiae. These yeasts produce enzymes that catalyze the reaction that converts the substrate into energy for the yeast and also into alcohol. So during the production, the fermentation yeast produces enough enzyme needed for alcoholic fermentation. So these enzymes will continue fermenting far beyond even the need for the yeast, for the energy for the yeast, even longer after the production has stopped. The enzymes continue, they continue fermenting. And so because of this particular fact on this property is what is exploited industrially while carrying out fermentation. So after this, like I mentioned to you, that the product, everything is modeled together. So in this case also of ethanol fermentation, everything is modeled together, so you have to carry out the separation. And as I listed for you, several techniques, and distillation is one of them. So in case of ethanol, it is distillation process that is used. So it depends, in this process depends on the quantity of beer handled, the purity of the products required, and then the degree of heat economy being exercised. So carry out the distillation based on these factors. So conventional process consists of at least 2 distillation columns. I mean, the process of ethanol distillation, it consists at least 2 distillation columns. One is in the beer or stripping column. The beer or stripping column. In this case, the alcohol is stripped from the beer and then concentrated to between 50 to 60% volume by volume. Then the beer is then preheated by hot stillage coming from the bottom of the column, of this stripping column. Then the other column is rectifying column. In this case, the alcohol is concentrated to about between 95 to 96.5% volume by volume. Then it is withdrawn from the top of the column. The vapors at the top, which consist volatile components like aldehydes, are condensed and some returned as reflux. Fuel oil, which contains, among others, the fuel oil contains several compounds, amongst which you have amyl alcohol, is withdrawn from the lower part of the column. So ethanol or ethyl alcohol is a very important product where you can have several derivatives, several compounds that are very useful, organic chemical industry. Such are diethyl ether. You can get your diethyl ether by reacting that alcohol with concentrated sulfuric acid at about 150 degrees centigrade. And this diethyl ether is used as a solvent. Then another derivative from ethyl alcohol is ethylene. You obtain this by dehydrating it using concentrated sulfuric acid. You dehydrate the ethanol using concentrated sulfuric acid at temperature of 170 degrees centigrade. And this is mostly, ethylene is mostly used in polymer applications like polyethylene. It can also be used as other chemical intermediates. Another product you get from ethyl alcohol is ethyl chloride, which you obtain by reacting the alcohol with hydrogen chloride. You also have butadiene, In this case, this one is used for making synthetic rubbers. And you obtain your butadiene by reacting the alcohol with acetaldehyde. Another product you will get from the ethyl alcohol is acetaldehyde itself. You get this by dehydrating the ethyl alcohol, and it gives you acetaldehyde. It is used as a precursor for, acetaldehyde is used as precursor for acetic acid and also for acetic anhydride. But both acetic acid and acetic anhydride are used for making acetates such as cellulose and vinyl acetates. Cellulose acetate is used in synthetic fibers, while vinyl acetate is used in polymers. So you have your polyvinyl acetate, and also is used also in paints. So the acetaldehyde can be used to make also normal butyl alcohol, which is a very useful chemical intermediate. And then following this equation, reaction equation, this is for these processes, this is what happens. This is how you get your N-butyl alcohol from acetaldehyde. So we have advantages and disadvantages of fermentation process. So one main advantage is that you have renewable resources to use in fermentation. It's not something that is exhaustible. You have renewable, you can plant the raw material and so on. So, and they proceed under mild conditions. That's another advantage. So despite even these two advantages, there are some disadvantages that make it difficult for fermentation process to continue thriving. So such is that the production of sucrose, the disadvantage that the production of sucrose and starch require a use of large area of land for you to plant, and also a lot of labor is also involved. So this will make the production of your raw material very expensive relatively. Then another disadvantage is that fermentation is a wasteful process, wasteful of raw material. If you want to produce 1 ton of ethanol, you will waste 1 ton. You also produce 1 ton of carbon dioxide. So for every ton of ethanol produced, 1 ton of hydrogen peroxide is produced from that raw material. And so it's really wasteful. And also another disadvantage is that it's very slow. Process is very slow. It has to be carried out in dilute solutions. For instance, the ethanol fermentation, it takes about 2 days to take place, containing just very dilute ethanol, 9%. So as a consequence of this, you need large volumes of reactors, and then to carry out this in large volumes, and then its separation cost is high. So these 3 disadvantages that I've just listed are the things that make fermentation route to organic chemicals very unattractive, especially in highly developed countries, except for products that you cannot obtain through any other process except this fermentation. So such product like citric acid is practically all made by fermentation. The only other source of your citric acid is just by extracting directly from the citrus fruit. So that's the situation. And this brings us to the end of this lecture, and the end of my section of ICH 311 with you. And we will see what we will do next and we'll plan maybe some tutorials and then some tests. So I don't know if anybody has any question. At this moment, I want